Question for you. If all of our cells contain the exact same DNA, what makes a brain cell so different from a liver cell, from a bone cell, from a muscle cell, or a skin cell? We did begin as one single cell with 23 pairs of chromosomes that began dividing and dividing till there were multiple cells that now don't look the same anymore. So what happened? Well, the answer to that eventually leads to one of my favorite biology topics, epigenetics. But that's another video. I promise I'll do another video on epigenetics later. This video is called Transcription and Translation for a reason, and I have to cover transcription and translation first because it provides the foundation to understand all that really fascinating stuff that comes later. So, back to my first question. Why is a skin cell physically, functionally different than a bone cell when they both contain the exact same DNA? Good question, yeah? Well, if you think about it, how much information is actually contained in the full set of chromosomes that each cell has? It's enough information to hold over 20,000 genes, more than that, but most of our DNA doesn't code for genes. Anyway, can you imagine if all 20,000+ genes were expressed in a single cell? How kooky and confused would that cell be? It wouldn't work because, you see, each of our cells only uses a small fraction of our genes at any given time. So most of our genes are not turned on in any given cell. Most of our genes are turned off. And the select handful that are turned on determine what kind of job that cell is going to do. And it is through the process of transcription and translation that determine which genes get turned on, expressed, or not expressed. Transcription is making a messenger RNA from a DNA template, and translation is taking that messenger RNA to a ribosome where it can get translated into a protein.
It's very simple when you look at it like this. When DNA replicates itself, that's called replication, and this would occur when the cell is getting ready to divide. It would need a second copy of its DNA in order to divide. Messenger RNAs are used as instructions for protein synthesis. Messenger RNAs are copied from DNA, and this copying of the messenger RNA from DNA is called transcription. When that messenger RNA gets translated into a protein, that's called translation. And this occurs on ribosomes. So replication is DNA synthesis, transcription is RNA synthesis, and translation is protein synthesis.
I just want to quickly go over some terminology which you may already know. Amino acids are the building blocks for protein, and there are 20 of them. And a string or a chain of amino acids is called a polypeptide. When that polypeptide gets folded, it becomes a functioning protein. Sometimes the terms polypeptide and protein get used interchangeably. Don't get confused by that. A folded polypeptide is a protein. And a DNA or RNA sequence of 3 nucleotides long is what's called a codon, and an example is ACG or GUA. An anticodon is the 3 complementary. Okay, that should be an E, not an I. It's complementary, not complementary. Not a big deal either way, but now you don't have to make the same mistake I did. So, yeah, the anticodon is just the 3 opposite base pairings to the codon. For example, if the codon was GUA, the anticodon would be CAU. The codon is on the messenger RNA, and the anticodon is on the transfer RNA. Oh yes, there's another RNA called a transfer RNA, and it's better if I show you a visual depiction of that instead of a definition. Alright, so here we have our messenger RNA carrying the protein instructions, which it copied from the DNA in the nucleus and then it exited through the nuclear pores, one of the nuclear pores, to the cytoplasm, and this is where the messenger RNA now has to connect with a ribosome before translation can occur. And the other kind of RNA molecule you should be familiar with is the transfer RNA, and this is it here, the funny looking T-shaped molecule. Here's another one. A transfer RNA will carry an amino acid that corresponds to the messenger RNA codon, and this recognition occurs through the pairing of the codon with the anticodon on the transfer RNA. That's an anticodon. So if the codon was CAG, like you see here, for example, the corresponding anticodon would be GUC. And as more amino acids get added on, this polypeptide chain grows. That's what these colorful balls represent, is a polypeptide chain. Transfer RNAs often are shown looking a bit like a T structure, so maybe that can help you to remember that it's a transfer RNA. So transcription RNA synthesis is occurring here in the nucleus, while translation protein synthesis occurs here in the cytoplasm on a ribosome. Of course, this is a eukaryotic cell we are talking about, where we have a nucleus, and all of this would occur in the cytoplasm in prokaryotes. By the way, ribosomes contain another type of RNA called ribosomal RNA in this complex here. So we've actually got three types of RNAs: messenger RNA, transfer RNA, and ribosomal RNA. Now, if you're ever asked to generate a messenger RNA code from a DNA code, you would pair A's with U's, T's with A's, G's with C's, and C's with G's. Here's an example. If we were given a string of DNA-based triplets like so, we could easily convert that into a messenger RNA and then the corresponding anticodons and amino acids. So let's do that. C pairs with G, A pairs with T, G pairs with C, and so on. Now, if this were a complementary DNA strand, this would be correct. However, this is not a DNA, it's an RNA, and ribonucleic acids do not have thymine. Instead of thymine, RNAs have uracil. So wherever you would want to put a U, wherever you would want to put a T, put a U instead. Then, if you're asked for the anticodons that would match this segment of messenger RNA, it would be the same, but wherever you see a U, you would pair that with an A. Don't pair U's with T's. That's a common mistake. And that would be our anticodon. Now, to find out which three amino acids are coded for here, we would look at the messenger RNA codons and refer to either a codon wheel or a codon chart. And they're both easy to use. On the wheel it tells me GUC would give us the amino acid valine. You start in the middle with G, move your way out to U and find C, and there's valine. UGG gives us, start in the middle with U, outwards to G and another G, and there's tryptophan. So our first two amino acids is valine and tryptophan. Notice notice that to find the correct amino acid, we're using only the mRNA codon. We can't use the DNA base triplets or the anticodon. It doesn't work for either the chart or the wheel. We can only use the messenger RNA codon to get our amino acid. But if you're given either the DNA or the anticodon, know that you can translate in any direction. Now let's use the codon chart for our last amino acid, CGA. So you can see it's just as easy to use. So for the chart, we line up our first base and our second base. Our first base is C, second base is G, so you line up those two columns. We're in the square, and then you find the last base within the square. CGA is there, tells me that we're going to get arginine. Notice we can have multiple amino acids multiple codons coding for the same amino acid. So CGA gives us arginine. Sorry, my elbow does not bend that way. And you should get the same result whether you use the codon chart or the codon wheel. You should get the same amino acid. And there are our three amino acids, valine, tryptophan, and arginine. So we transcribed here, going from DNA to RNA, and then translated from RNA to amino acids. Transcription, and then translation. And remember, a chain of amino acids gives us a protein. And that's it for transcription and translation. It was a brief overview, but hopefully now you have a good grasp on the general idea, and maybe you can describe it in your own words. That's how you know if you really understand something, is if you can describe it in your own words. So hopefully you can do that.